Hello everyone, welcome back to my channel Competitive Botany. In this video, we are going to discuss non-allelic gene interactions. In the previous video, we are talked about allelic gene interactions. If you not yet seen that video, please go through that video for better understanding. Okay, let's see non-allelic gene interactions. Non-allelic gene interactions are nothing but interaction between alleles of the two different genes or non-alleles. Look at the picture carefully. Capital A, capital A. So these two are the homologous chromosomes as they contain two same alleles, similar alleles. That condition is nothing but homozygous condition. They belong to the same gene. Remember. So again these two capital B, they belong to capital B are the two alleles belonging to single gene. So here we are observing the interaction between two alleles of two different genes. Okay. So interaction between alleles of two different genes, that is nothing but non-allelic gene interaction. Okay. So non-allelic gene interaction is of following types such as epistasis, complementary genes, supplementary genes, inhibitory factor, duplicate genes and polymorphic gene. So let's see one by one. Okay. So first one, epistasis. Let's see the first one, epistasis. Epistasis is nothing but the expression of one gene or gene pair hides or masks the expression of the other non-allelic gene. When one gene masks the expression of other non-allelic gene, it is nothing but epistasis. Okay. When one gene masks or hides the expression of another gene, here another gene is nothing but non-allelic gene. Remember, that is nothing but epistasis. Let's see. The gene which suppressing the other gene is nothing but epistatic gene. The suppressed gene, so it is suppressed by another gene. The suppressed gene is called as hypostatic gene. Okay. So epistasis is again of two types: dominant epistasis, recessive epistasis. If the dominant gene masks the other gene, that epistasis is nothing but dominant epistasis. If the recessive gene masks or hides the expression of another gene, that is nothing but recessive epistasis. Let's see in detail. First one, dominant epistasis. Here the dominant gene masks the effect of or expression of other non-allelic gene. So dominant gene masks the effect of other gene. That's why this is called as dominant epistasis. Okay. Let's see the example for dominant epistasis. So fruit color in summer squash cucurbita is the best example for dominant epistasis. Here the fruit color is governed by two genes. So the dominant gene W, which is meant for white fruit color, it suppresses the expression of Y, capital Y gene, which is responsible for yellow fruit color. Remember, capital W gene expressing white fruit color. Of course, capital Y gene expresses the yellow fruit color. But they come together or they interact together when they present in single organism, white color expressed. Why? Because here capital W is the dominant gene which suppresses the another dominant gene. That's why this interaction is called as dominant epistasis. Dominant epistasis. Let's see the example in detail. So look at the grass. When a pure breed white fruit color plant crossed with a true breed green color fruit plant, it will give, male will give capital W capital Y gamete, female will give small w small y gamete. When they fuse together, resulting in F1 individual. F1 individual is heterozygous for both the genes. So that's why it is having 4 different alleles. Remember? Okay. So it is also expressing a white color. Why? Because capital W is for white color fruit, which suppresses the yellow fruit color. Remember, which is a dominant gene, it is suppressing another dominant gene. That's why this epistasis is called as dominant epistasis. When the F1 plants allowed to self-pollination, when they allowed to self-pollination, resulting in the formation of 16 types of progeny. 16 progeny will obtain in the F2 generation. Okay, so have a look. Wherever you find capital W and capital Y, only white color will expressed. Why? Because here you have to remember the conditions. Capital W, capital Y, when they both are present, capital W suppresses the capital Y. That's why white phenotype produced. When capital W and small y present, here you have to place either capital W or small w. Okay. Here also the same condition. Why? Because here also white color expressed, because capital W is the dominant gene which is expressing white color, white fruit color. The third condition, small w and capital Y. In this blank you will fill either capital W, capital Y or small y. Anything it can be. The color is yellow. Why? Because capital Y gene meant for yellow color only. There is no capital W gene. That's why yellow will expressed. Okay. So remember. So last condition, when capital W is absent and capital Y is absent, they express green color as parent. They express green color as parent. Okay. So let's see the Punnett board. Wherever you find capital W, all are getting white fruit color. Wherever you find capital W is absent, they are showing yellow fruit color. If capital W, capital Y, both the dominant alleles are absent, the fruit is in green color. Okay. So here we are getting the ratio of 12:3:1. 12 white progeny, white color fruit progeny obtained, 3 yellow fruit colored progeny obtained, 1 plant obtained with the green fruits. 1 plant obtained with the green fruits. Okay. Let's see the next type of interaction, nothing but recessive epistasis. So if a recessive gene does not allow the expression of other non-allelic gene, is called recessive epistasis. Okay. So if recessive gene suppresses the expression of other gene. Okay. So if recessive gene masks the or hides the expression of other gene, that epistasis is nothing but recessive epistasis. Here coat color in mice is the example. Okay. So the coat color of mice is controlled by two pairs of genes. Here, the dominant gene capital C produces black coat color, whereas capital C is, when capital C is absent, that means small c is present, it will produce albino color. Okay. So whereas capital A produces agouti color. Capital A produces agouti color in presence of capital C. When capital C and capital A present in single individual, they produce agouti color. Okay. So A cannot express in absence of capital C. That means if there are two small c's or if there are two small c genes, the resulting individual is in albino color. That means recessive allele small c is the epistatic to dominant allele. Okay. So remember the situation. You have to be remembered carefully. Okay. So let's see the Punnett board. So when the pure breed capital C small a black individual is crossed with small c small a. Remember, here small a is capital A is absent, small a is present. That's why capital C produce black color. Here capital C absent, small c present, it suppresses the agouti color. Dominant gene, recessive gene small c suppresses the agouti color. Okay, that's why it produces albino color. So when one black individual crossed with an albino individual, so when the capital C and capital A come together in F1 individual, they will produce agouti color. When they come together in an individual, they produce agouti color. Remember, when they are allowed to inbreed, they will produce 16. There is a chance to produce 16 progeny. Have a look. See the Punnett board. Here are the combinations. Wherever you get, here you have to be remembered carefully the situations. What are the situations? Let's see. If capital C, capital A present in single individual, it will produce agouti color. If capital C only present, capital A is absent, then it will result in the black color. If capital C absent, capital A present, that will result in albino color. If small a individual obtained, that is in obviously albino color. Okay. So let's see the Punnett board. Wherever you find both the alleles in single progeny, you have to be remembered that agouti color will be produced. How many agouti will be produced here? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. So 9 agouti progeny will be produced. So how many black colored individuals are there? 1, 2, 3 black colored individuals. Again 4 albino individuals are obtained. So the ratio, the recessive epistasis ratio is 9:3:4. 9 agouti will be produced, 3 black colored produced, 4 albino colored individuals produced. So this is about recessive epistasis. Let's see the non-epistatic gene interactions. Okay, coming to the non-epistatic gene interactions, first one is complementary genes. Complementary genes are nothing but certain characters of organisms are controlled by the interaction between two or more genes occupying different loci and they inherited from different parents. Remember, they are complementary to each other. Certain characters of organisms are produced by the interaction of two or more genes occupying different loci and also they are inherited from different parents. Remember. So as they are complementary to each other, when they come together and all together, new phenotype expression is produced. When they come together in F1 individual, for example, I am saying they will produce a new phenotypic expression. Okay, so in the progeny, because they are complementary to both. Okay, that's why they produce a new phenotype when they come together in an individual. Remember. So let's see the example. Flower color in Lathyrus odoratus. It's nothing but sweet pea. So in sweet pea or Lathyrus odoratus, capital C, capital P genes are required in sweet pea to produce anthocyanin pigment. So due to this anthocyanin pigment, they will produce purple color flowers. They will produce purple color flowers. But in absence of any one gene, that means in absence of either C or P, they definitely do not produce purple color flowers. They will result in white color flowers. Okay. So capital C, capital P genes are complementary to each other for the production of anthocyanin pigment. Results in the formation of purple color flowers. Okay, so let's see the cross. When pure breed white color plants are crossed with again white color plants, so they are having different genotypes even though they are producing white color. Why? Because here there is no capital P to produce anthocyanin pigment. Why? Because if anthocyanin pigment is produced only when presence of two genes. If the two dominant genes present in an individual, they will only produce anthocyanin. Here capital P is absent, here capital C is absent. That's why they produce white color flowers. Okay. So when they crossed together, they'll result in F1 individual having both the genes. F1 individual having both the genes. So that's why they are in purple color. Why? Because they produce anthocyanin pigment results in purple color. Okay. That's why the F1 hybrid in purple color. Okay. Let's see F2 progeny. So when the F1 plants allowed to self-pollination, F2 progeny will be obtained. Here also 16 combinations of plants are obtained. Let's see. Let's see the Punnett board. So these are the situations you have to be remembered. Okay. So when capital C, capital P present, it will produce purple color. When only one gene is present, it will produce white color. As the parent, if both the genes, both capital genes are absent, then results in small c, small p. So it will also produce white color. When the capital C and capital P present, then only the flowers produce purple color due to anthocyanin pigment formation. Remember. So look at the Punnett board carefully. These are all purple colored flowers. Why? Because all are having, that means 9 plants are having both capital C and capital P capital C and capital P alleles. That's why they produce purple color. If wherever you find only capital C there is absence of a capital P, or only capital P is present, small capital C is absent here. So these are all the situations the plant produce white colored flowers due to absence of anthocyanin pigment. Okay, so here the ratio is 9:7 ratio. 9 plants are produced with purple color and 7 plants are produced with white color. Okay, let's see the next type, supplementary genes. Sometimes the interaction involves two different types of genes in which one type expresses itself independently but other has no expression of its own. When such two genes interact, they will produce a new character. Okay, so listen carefully. When the two genes interact each other, they will produce a new phenotype. Among them, one will able to produce the independent expression, the other is not able to produce independent expression. Remember, for this, the skin color in rats is the example. Same example we have seen in recessive epistasis. So same class only we are going to deal. Okay. So in rat there are three common skin colors or coat colors. They are nothing but black, albino, agouti. Agouti is the wild phenotype. So black color is governed by dominant gene C. When A and C are interacted, they will produce agouti color. A does not show any visible effect if the capital C is absent. Okay, A does not show any visible effect if it is alone. If A is present alone, it will not produce any phenotype. But it will produce with capital C. It will present with capital C. It is able to produce agouti or wild phenotype. Remember, let's see. But the gene C is able to express itself independently. C can express itself independently, but A is not expressed independently. A is not able to express independently. It will only express when it is present with capital C. Remember? Okay. So let's see the example, or let's see the class. So there the pure breed black rat, nothing but having the genotype of capital C, capital C, capital A, capital A. Look at this. Here there is no capital A. That's why capital C is able to produce black coat color in rat. Here capital C is small c, and capital A, capital A. It will produce albino color. Why? Because small c is present. It will produce albino color. When they produce the gametes, they will fuse in F1 hybrid. They will fuse to form F1 hybrid. F1 hybrid having the genotype capital C, capital A, small a. Look at this. Observe carefully. Here two genes are present. When the two different genes come together, they will produce a different kind of expression. They will give a different kind of coat color, agouti. Okay. So F1 individuals all are having agouti. So let's see, when the F1 individuals are allowed to inbreed, that means one F1 rat is crossed with another F1 rat, resulting in the formation of 16 kinds of progeny. There is a chance to get 16 kinds of progeny. Look at the Punnett board. So these are the male gametes, these are the female gametes. They will fuse as shown in the figure. Okay, so if capital C, capital A present, they will always produce agouti color. If capital C alone present without capital A, they will produce black color. If small c present, they will always produce albino color. So here the situations you have to be remembered. If capital C, capital A present, they will produce agouti color. If capital A is absent, both small a is present, black color resulted due to capital C is able to produce black coat color in rat. Let's see the third situation. So there are two recessive genes, small c present with capital A or capital A small a. It results in the formation of albino coat color because A is not able to show any kind of expression. There is no C, there is no capital C, that why small c always produce albino color. The fourth situation, small c, small a always produce albino color. Okay, so the phenotypic ratio is 9:3:4. Agouti 9, black 3, albino 4. Okay, this is the Punnett board. You have to understand carefully. Thank you.